Hi guys, in an up-and-coming video, I wanted to cover T cell types, right? T1, T2, T17, for example. And I realized to do that, I really need to give a background overview of how the immune system fights pathogens in general. It's going to be sort of collating all the information that I covered in previous videos, as well as bringing in some new information about how parasites and cancer might be attacked by our immune system too. So let's just jump into it. This image here is what's called an ELISA spot, and in the next video I'm going to go over some of the details of it. It's just a very cool and beautiful image right now, but in the next video I'll break it down on what's happening in an ELISA spot. So first of all, there's more than just bacteria and viruses out here. There are, in fact, intestinal worms, as an example. There are multicellular organisms that infect you. And this here, right here, is what's called a pinworm or a threadworm. And it affects about 11% of people in high-income countries. So if you're in Australia right now, there's a 1 in 10 chance that you have these worms in your intestines right now. But it gets as high as like 60% in low-income countries, in which they struggle a little bit more with clean water and everything like that. So it's a very prevalent disease. In fact, pinworms is one of the highest prevalent diseases in the world. It's massive. Its symptoms are tiredness and irritability, which we kind of all suffer from a little bit sometimes, but a major one is itchiness. Now this itchiness, I'm going to get a bit graphic here. This itchiness is caused by the female worm poking its head out, typically at night, of your rectum and laying eggs around your rectum. And the life cycle goes, you then itch it and get eggs on your fingers, which then either get into the water or on food, and then they get into someone's mouth. Very, very gross. But the point is that there are some peculiar organisms that we're infected with, and they are incredibly common and often overlooked. But let's just break down how might the immune system respond to this. Well, if we look, here we've got a macrophage, and it's come across some bacteria. Now, what that macrophage might do, macro means big, phage means eater, is it will eat those bacteria. There are a number of ways that it can get into the cell. Bacteria are typically phagocytosed, which is often aided by complement proteins or antibodies. They might macropinocytosis, which is when the membrane crawls around the pathogen and then sucks it in. Typically, these smaller protein-mediated pathways are for viral particles only, because only small bites can be made. But this is typically what a macrophage will do when it comes across a small pathogen like a virus or a bacteria. It will eat it. But let's have a look at this worm. Now, already we can see this macrophage is going to struggle to eat that worm, but the phagocytosis is made even more impossible when I show you it to scale. So this is, you can't even see it anymore, but this is a macrophage and a pinworm to scale. So they're now at the appropriate sizes on the same scale bar. And you can see that you almost can't even see the macrophage there. It's just there. There's no chance for that macrophage to eat that pinworm. So obviously we're going to have to deal, the macrophage has to deal with those 2 or 3 different kinds of pathogens differently. And so here's the key thing here is different pathogens require different immune responses. So I'm going to actually do a breakdown on this and try to give you a full panel breakdown. This video might take a wee while, and you might need to go over the video a few times to really soak it in. But how does the immune system deal with each of these problems? So we've got viruses there, of course, they're tiny on a nanometer scale. We've got bacteria there, which are a little bit, well, quite considerably bigger than a virus. Then we also have cancer cells. So this is when our own cells begin to mutate and divide in ways that they shouldn't. And then we have parasites, and in here I'm just going to look at the pinworm kind of parasites here. And so these are eukaryote cells. These are made up of similar cells to what we're made up of, right? And so the immune system has to deal with them a little bit differently, because bacteria are more unique from us than a pinworm or a threadworm. They're more similar to us. So our immune system has to work a bit harder to sniff out a pinworm than it does a bacteria. So let's just jump into it. What are some of the ways that the immune system might fight these? Well, there's the antibody responses. Now, I went into this in detail in an antibody video, but just quickly, we've got neutralization. That's when antibodies bind to proteins on the surface of pathogens essential for the function of that pathogen. We've got agglutination and precipitation, which is essentially when antibodies bind multiple pathogens or toxins, and by binding multiple of them, it clumps them together. And that prevents them from spreading. It makes them easier to phagocytose, and it can precipitate them out of your liquids, your plasma, your blood, your extracellular fluid. It can precipitate it out, and so now those pathogens or toxins can't do more damage. Now, agglutination is for pathogens, and precipitation is for toxins because it precipitates the soluble chemicals out of solution. Next, we've got complement recruiting. So this is an antibody function again. So antibodies often bind to proteins and compounds on the surface of pathogens, and then they bring in complement proteins, which are innate immune cell proteins, innate proteins that float around your blood and have the ability to attack and kill pathogens, but also recruit immune cells to those pathogens. Then we've got cell lysis, and this is done majorly by two main cells in your body, the cytotoxic T cell and the natural killer cell, and that's when they will pop a host cell using things like perforin, which is a protein that creates a pore in the membrane of the host cell. Then we've got phagocytosis, that's when a macrophage or a neutrophil eats up a pathogen. Then we've got neutrophil degranulation or netosis. This is when essentially neutrophils secrete out and release a whole bunch of cytotoxic compounds or enzymes that produce cytotoxic compounds like bleach, but also like proteases such as neutrophil elastase. And then we have an interesting one. We've got a vascular anatomical, right? So this is a bit more of a macro response, but an example is the vasculature in your nasal cavity becoming more permeable, so fluids flood out of it, creating snot and mucus. Similar things happen in our gut, where we get fluid flushing out into our gut, and actually our gut undergoes enhanced peristalsis, which is the process of pushing liquid out and food out of the intestine. I shouldn't say food, but fecal matter out of the intestine. And so those are actually immune responses. So we if there is a pathogen in our gut, we might pump a whole bunch of liquid into it, and we might accelerate the peristalsis of the intestines, which creates diarrhea, and that helps flush out the pathogen. So it's sort of this macro response, and it might benefit in some diseases more than others. So now let's compare how these immune system responses, how effective are they against these different threats to our body, these different diseases. So neutralization, that's excellent for viruses. So the antibody coats the virus, preventing the virus from entering cells. It's pretty good for bacteria. Some bacteria are actually intracellular bacteria, and so it can help block the effector proteins on the surface of the bacteria, but it's less effective, this process. On cancer cells, there's not a lot to neutralize because a lot of what drives cancer is self proteins, right? So there won't be an antibody that will bind to a self-growth protein, because then it will bind to all the proteins in your body. So neutralization is not very effective for cancer. And there is some evidence that it's reasonably good against pinworms. It coats the pinworms, preventing the pinworms from doing what they do. Some of what they do is they trick our immune system. And so by deactivating some of the proteins on the surface, you can deactivate the response by the parasite in order to fight our immune system. It's part of the battle there. Agglutination and precipitation, fantastic for viruses. Grab those guys, pull them together, make them more consumable, make them less effective at spreading. Really good for bacteria. Again, pull them together, disable them. Not really applicable to cancer cells again, and not really applicable to worms, right? If you saw how big the macrophage is to the parasite, imagine how big the parasite is to an antibody. There's no way a tiny little antibody is going to be able to pull two worms together, right, and have the strength to hold them. That's impossible. So now we've got complement recruitment. A big part of that is the membrane attack protein. The membrane attack protein pokes holes in the membrane, which can lyse the pathogen. Now, some viruses, this will work because they have a membrane, they have an envelope. Other viruses don't have an envelope, so that absolutely won't work in that situation. Bacteria, this is what really it was designed for, the complement recruitment. Grab onto that bacteria and puncture a hole through it to lyse it and kill it. Yep, so that's fantastic for that. Cancer, again, not that applicable. I couldn't find any research that suggests complements kill cancer. In fact, I found some research that complements helped cancer, and I'll go into how that could possibly be the case a little bit later on. And there is some good evidence that complements can help disable, attack, and kill parasites. So this is a winner for that one. Now we keep going down the list. I know this is a lot of information and you're going to have to go over it a couple of times, but go through the slides as well. It's certainly a great review thing. Maybe create flashcards for it if you want to really memorize what's going on here. Okay, so next up we've got cell lysis, so cytotoxic T cells and natural killer cells. This is what they were made to do, right? Detect a virus inside the cell, kill that cell so the virus can't keep replicating, right? So this is really what those cells were doing. So that's 3 stars there. But there are some intracellular bacteria that proliferate inside of cells, and in that case, these cells are helpful, but it's certainly more of a viral thing than it is a bacterial thing. Cancer cells, heck yeah. So this is actually one thing where the natural killer and the cytotoxic T cells excel at. They recognize the mutated proteins in the cancer cell, or they don't recognize it in the case of a natural killer cell. They don't recognize that as a self-antigen, but they recognize that there is mutations going on in the cell and they pop it, right? So that's right in the wheelhouse of the cytotoxic T cell and the natural killer cell. There is no research on this. I looked and looked and looked for hours, but I would love to know, does the perforin, the cell popping compounds of the TC cell and the NK cell, can they attack and kill a parasite? And I think that they could, but the experiment that I wanted to see, which was a genetic deletion of perforin in mice, which are then transfected with these sort of macro parasites, these multicellular parasites, what happens, hasn't been done. And so if researchers out there, I'd love to see you jump on that one. I'm an innate immunologist, so I'll stay clear of that. I like macrophages and microglia, but yeah, any sort of NK or TC cell researchers out there, jump on that question for me. Phagocytosis, it's less of a viral thing, right? The viruses are actually super tiny and they spread throughout the liquid, your bodily fluids so incredibly quickly that there isn't enough eating power in your body to make a huge difference with phagocytosis. Macrophages, of course, do phagocytose viruses, and then they display their antigens and induce an antibody response through the adaptive immune system, through T cells and then B cells. So in that respect, phagocytosis is really important in fighting viruses, but it's not directly important. It's not the eating of the virus that's massively important in killing those viral particles. Whereas bacteria, phagocytosis has a much bigger role. And there's been some great studies where if you block neutrophil phagocytosis, you can end up with rampant bacterial infections in animal models, of course. So the phagocytosis pathway is actually a key antibacterial pathway for neutrophils and macrophages. Cancer cells, there is some evidence here that phagocytosis does occur in cancer cells. Essentially, there is a signal from the cancer cell. There's a number of potential cell surface signals there that tell, if the cell mutates, it tells a macrophage, kill me and eat me. And so that's an important system in our body for preventing cancer development. So phagocytosis does have a role in eliminating cancers. And of course, it has no role in getting rid of those 10mm parasites. You know, they're this big, and your macrophage can't even see, so there's no chance that a macrophage could eat one of these giant worms. Neutrophil degranulation in nets, it's less of a deal with viruses. Again, it's that problem of diffusion all throughout our bodily fluids. There's so many there that you couldn't possibly, it would be very difficult once the disease is starting to divide. It would be very difficult to bleach out enough of an area, bleach out your entire lungs if you've got SARS-CoV-2, or you protease out your entire lungs if you've got SARS-CoV-2 to get rid of those viruses. Perhaps in the very, very early stages of an infection it helps, but there's not a lot of evidence that it helps massively in the later stages. If anything, it seems to contribute to pathology because now you're starting to get that collateral damage. Whereas bacteria, yep, that's fantastic. You know, bacteria typically, they grow from a site and they expand and expand and expand. That's much easier for the neutrophils to come in and just nuke that area. It's a local infection. Let's nuke that local infection with our non-specific cytotoxic compounds. Let's really kill them. So bacteria, yeah, that's one of the targets. Cancer, again, it seems more detrimental than it is positive, right? And we'll go into a little bit in the next segment as to why that is. It's sort of all tied together and why some of these things actually make cancer worse rather than better. But parasites, yes, it's great. Again, you've got this local thing happening. So if neutrophils surround the worm and start attacking it with digestive enzymes and reactive oxygen species, it can attack and kill that. So that's a fantastic way to get rid of that parasite. Last but not least, the vascular anatomical responses. So there is a little bit of benefit in the vascular anatomical responses when it comes to viruses. There's some evidence that diarrhea is a way to get rid of gastrointestinal viruses, and that the snot that we produce is relatively effective at getting rid of the nasal viruses, although there is also some evidence that drugs that block the snot production, like pseudoephedrine, actually help reduce viral load in things like influenza. So it's a little bit up in the air. It's not as important response compared to other pathogens. Bacteria, yes. If you get, if you think about you've eaten something wrong, it's got a lot of Campylobacter into it. You've eaten some raw chicken, maybe full of salmonella. Your body might recognize that and immediately initiate a massive anatomical response of peristalsis of the intestine and secretion of mucus and fluids into your gut. And you might have 24 hours of food poisoning, right? So you might have that very quick response that is quite beneficial at eliminating those bacteria. Cancer, we've got a double negative here, and this is where I was talking about how neutrophils and, say, complement proteins are actually might be detrimental in the case of cancer. There is a lot of evidence that inflammatory processes actually allow tumor development because many of the inflammatory cytokines produced by neutrophils or macrophages present in the tumor actually encourage the growth of blood vessels. So if you imagine you've suddenly got these dividing cells, there is no capillary inside that tumor to provide it with oxygen and to provide it with glucose. And so you can end up with a necrotic core inside of tumors because they don't have a blood supply, a hypoxic core inside that tumor. But if there's inflammatory signaling going on through complement proteins or neutrophils or macrophages, those inflammatory signaling molecules actually encourage blood vessel growth into that tumor, providing them with oxygen and glucose and taking away the waste. So now the tumor can grow and grow and grow because it's getting proper blood supply into that tumor. So that is why some immune responses are actually really detrimental when it comes to cancer. And parasites, this is massive, right? There's really, really good research that shows if you get a lot of parasite eggs into your gut, you will have an immediate mucus and vascular and peristaltic response to eliminate that out and prevent that infection from happening. So yeah, this is really where these guys get involved and have a huge effect. Right. Yeah, and you can start to see really important differences here. Like these ones here, which you could probably loosely say are the inflammatory responses here, right? The phagocytosis, vascular, and neutrophil responses. are much more effective against bacteria and are reasonably effective against parasites and are not very effective against viruses, right? They're not very effective against viruses. So you can start to see that, you know, the adaptive immune system up here is kind of good against viruses and bacteria, whereas the inflammatory immune system is probably a bit more important when it comes to the bacterial response. And there's a bit, if you have a look down, there's a few more differences there. And so this is where it becomes important. Our immune system needs to be able to recognize and respond differently depending on what the pathogen is. And that is where a lot of these different cellular subtypes of T cells and other cells are important in regulating different kinds of immune response. So here's just an example. And this is probably most similar to a viral infection. So let's just say this is an influenza infection. So on day one, you get a massive spike in the influenza, then it gradually goes down, and eventually you manage to clear the flu. This would actually be really good. I'd be really happy to have got rid of flu by 14 days. So we've got time on the x-axis and magnitude on the y-axis. Now, if we look at inflammation, inflammation happens almost instantly. So we get this massive inflammatory response. Immediately we get a runny nose, and immediately we get a temperature, which is from those inflammatory cytokines signaling our body to up its metabolism. And yeah, so we see this inflammation, we see this gradually cascade. Now, if we look at the antibody response, it takes time. Remember, there's only a few cells in your body that might be able to recognize and create the appropriate response, including a cellular response or a humoral response to that influenza. So your body takes time for the antigen presenting cell to sample that pathogen, present the antigen to a huge number of T cells and B cells, T cells, B cells, have a look at this, have a look at this. Sorry, T cells, the antigens are floating around in which the B cells recognize. And so it takes a huge amount of time to mount this antibody response. And then as soon as we do, we really effectively tackle the disease. Once those antibodies start to spike up, we really effectively tackle those disease. So the important thing here is actually there's a temporal difference as well as a pathogen difference. So the different kinds of immune responses have different strengths in terms of both the pathogens that they respond to and the time. Now, this time thing creates a very interesting reaction with the immune system. So if we look at initial pathogen level, right? So this isn't we have an infection. This is say I snort up some viral particles, or I eat some chicken contaminated with a bacteria, or I get parasite eggs into me. If it's low or high, might depend on which immune system response is better, purely because of that time factor. So let's have a look. These antibody responses are fantastic if the initial contact with the disease is a low pathogen. We get inoculated, we get infected with a low level of the pathogen. And that's because at a low level, it's going to take time for that pathogen to mount a really devastating level of infection. And during that time, we're looking for our own adaptive immune cells that can respond to this and pump out antibodies to neutralize it. So if we have a low level of infection, we've got that time before we get serious pathological consequences to search for the cells that can produce, that have the appropriate T cell receptor or have the appropriate antibody that can deal with this pathogen. But if we get a bucket load of pathogen all simultaneously, that infection is going to be rampant and overtake our body, and perhaps we would even die before the antibody response could initiate. Perhaps, like, think about having a really contaminated salmonella raw chicken, right? You are going to get way sick before 7 days, right? And so if it takes roughly 7 days to develop that antibody response to it, and that cytotoxic response to it, you are going to be potentially dead before your body produce the appropriate immune response to it. So that's why we need these other cells. Now, Cytotoxic T cells are in the same state because they have that unique TCR receptor, so they need to be found by the antigen presenting cell, right? So there needs to be that there's only 1 or 2 cells that can deal with this antigen. We need to find them, they need to proliferate, they then need to go find all the cells and kill them, and we need antigen presenting and so on and so forth. So that takes some time. Natural killer cells are an innate immune cell that are always there, and they're just looking for any antigen that isn't yourself. So they can respond immediately. So that's why we get 2 ticks here for the high response here, but that's almost exclusively because of the natural killer cells there. The TC cells won't be helpful until later on the disease, and you might have died before the TC cells start to respond. Phagocytosis, purely because it can struggle to deal with high pathogen numbers, is better when it's dealing with low pathogens than high pathogens. When you think about, imagine having, you know, 1 trillion viral particles flowing all throughout your body and your plasma, is phagocytosis going to be able to clear those effectively? Not really. Neutrophil degranulation is and vascular responses is when our body really goes, I cannot deal with this pathogen in a specific response. I need to kill everything here now because I have found a huge amount of pathogens all simultaneously. So let's say you breathe in a massive load of SARS-CoV-2, your immune system is going to go, whoa, 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 let's get neutrophils there immediately and have a massive neutrophilic response to try deal with it. And it's one of those things that hopefully the neutrophils can get it back down to the low level, which will give our immune system time to respond. And I mentioned that, you know, if we get a whole bunch of parasite eggs simultaneously into our intestine and gut, we can initiate a very quick inflammatory response and a peristaltic response and a mucus secreting response to get rid of the eggs of that pathogen as fast as possible, of that parasite. And if we go back to, let me just go back to this graph here, you can see the inflammation, and this is a theoretical graph, but there are graphs that look a lot like this with real data. The inflammation has caused an immediate sort of minor drop in the pathogen, right? So the inflammatory response can help blunt the pathogen's infectivity and proliferation, giving your adaptive immune system time to produce antibodies. So this is a good example of a response. Often what happens though is that the inflammation can wipe it out before anything even happens. So we are zapping bugs all the time before a proper infection takes hold, and that's largely dealt with by the innate immune system because it's so quick at responding. So, you know, there might be a few viral particles here and they get zapped, and a few more viral particles, they get zapped, and then one starts to proliferate and our inflammatory system starts to go, let's just try contain this until our adaptive immune system can get in and really deal to it with antibodies. Right. So the key is different pathogens require different immune response, as well as different doses of pathogens require different immune responses, as well as different times in the pathology it requires different immune responses. So we need to be able to manage our immune system and draw on its strengths and draw on its weaknesses at the right time to deal with this range of pathogens, concentration of pathogens, and time frame of the pathogen infection. And that is why we have so many different kinds of immune cells. So here I've just got the T cell tree. This is very basic. It actually goes way more than this. But in the next videos, I'm going to be jumping into these groups of cells down here. The effector T helper cells that can be broken up into Th1, Th2, Th17, and Tregs. And we're going to be looking at perhaps, because each one of these types is better at managing a certain pathogen in a certain time point at a certain concentration than a different type. So we draw on the strengths and weaknesses of these cells to manage our immune system, because T helper cells are immune cell managers. They help the immune system. They sort of coordinate it. And so we need a different coordinator to coordinate a different kind of response to each different pathogen. And so that's coming up in the next video.